minutes left in class. Welcome. It's March 15th, 2021. It's Monday. It's the start of a lot of new things. It's the start of a new unit. It's the start of a new quarter, the last quarter of the school year. And it's also the start of a new week. So I'd just like to remind you all to remain as positive as possible. This is an opportunity to refresh, to feel rejuvenated, and to re-strategize. Think about what goals you're hoping to establish for yourself going into this last quarter of the school year. It's also an opportunity for reflection. So there was a survey that was handed out to you all virtually last week. It was a part of what's called the Semester 1 review materials module. So I would definitely encourage you all to take that survey. It gives me an opportunity to think about what went well and what I could do better to support you all, but it also gives you the opportunity to think about how you can step up your game in this fourth quarter. So I definitely encourage you, take 5 to 10 minutes to knock that out. And always, when you're at the beginning of something new, think about the opportunity you have to reflect before you jump into it. What do you want to change? How do you want to implement new strategies? How do you ask for help so that you put yourself in a better position? So these are things that I think are a great habit to establish when you're still in high school. And it could be something that you just start to do every week, something you could do at the beginning of every month, whatever, at the beginning of a school year or at the beginning of the calendar year in January. Whatever works for you, I think it's a good idea. But we do find ourselves here. We're starting a 6th unit of the class. We're officially halfway through biology now. This second half of the class is going to feel a little bit different. Number one, we've got a couple of really short units. This is one. Unit 6 is only going to last until Thursday. We're not even going to take a formal exam for Unit 6. We're just going to kind of transition right into Unit 7. And I think the second half of the class also feels different because it'll be more familiar to you all. We'll be discussing things that are probably a bit more interesting to you. Now that we've gotten some of the basics down, we're in a better position to jump into some more interesting and intriguing conversations, to talk about some new and cool technologies, to talk about evolution, which is something that I think always intrigues students. So I would be looking forward to the second half of the class if I were you. But of course, the second half is important because we are quickly approaching the EOC exam. So of course, remember that in order for you all to pass this class and get credit for it, not only do you have to get, at least the D in terms of what you do in class, but you also have to pass the EOC exam in May. So this is something that we take seriously, guys. And I don't want you all to just pass because, technically you could get a 3 and pass. What I want to see from all of you all is 4s and 5s. And I know that you all are capable of that, but we've got to step our games up, okay? So that is a good transition into my next topic of conversation. And you all can feel free to ask questions or, if there are any concerns, let me know. But obviously, you all should have the opportunity to check PowerSchool, see what you got in the third quarter, see what will be on that, what's it called, report card, as well as your transcript, and kind of take note of what went well and what did not. Some of you would have been in a great position because you did well on the test, but you didn't do any assignments, or you were missing 20 plus assignments. And that's what really hurt your grade. Some of you were really consistent with turning in all of your assignments, but your test performance was not where it should be. So find out exactly where you are and start to think about what is the strategy that is going to allow me to improve. Even if I got a B, there's still room for improvement. Even if I got an A, I'm not sure if anybody in this class got an A last quarter, but even if, regardless of where you find yourself, there's room for improvement. All right, and that's how I think you guys need to approach school in general. Some of you I know don't like school. You feel like it's a waste of time. You feel like you're not learning what you need to know for life in school. But school, like most other things in life, is a tool. It can give you what you want from it. If you go into it and you think I'm going to improve every day, regardless of what I'm given, then that's what's going to happen. But if you go into it with the mindset that you can't get anything out of it, that's also what's going to happen. Okay, so this is what I mean by thinking positively at the beginning of every week. So check your grades. Let me know. I gave you all as much time as possible to get things turned in. I didn't even submit the grades until about 3:15 p.m. on Friday, which was well after the deadline. So I was hoping people would be turning stuff in up until that last minute. See if that applied to you. I know some of you did try to work pretty hard up until that last minute, but just check. Obviously, everything that happened in that third quarter, the first quarter of the semester, that's done now. We're not worried about those missing assignments so much. I would love for you to try to do them just so you can get the practice, but it can't go into the grade book anymore. So we're starting fresh now. We've got what's called a tabula rasa, a blank slate. We're starting off with a blank slate. That gives you all the opportunity to say, hey, I may have had 30 missing assignments last semester. I may not have shown up for 20 different classes, as was the case with a couple of you, but I'm here now. And because of the grading policy, nobody has a grade lower than 50. So you're still in a position to pass the class. So we've got this blank slate. Take advantage of it. Make this quarter what you want it to be so that you can be in a good position to pass the class. All right. Unit 6 is all about biotechnology, and I will talk more about that. But today we'll talk a little bit about DNA fingerprinting. We want to know how it is used for comparison and identification of organisms. The essential question, what is my current level of understanding as we wrap up the third quarter? That's the wrong essential question. My apologies. All right, so I went over this briefly last week. It's probably a little bit small for those of you who are in person, so I apologize, but I think you guys are all in the call as well. So this is going to be the schedule that we follow through the end of the school year. Not end of the school year, through May 12th. May 12th marks the beginning of testing. So at May 12th, we're going to kind of transition into something else that they haven't released yet. But we've got this scheduled for essentially 2 months. What's that? Leave later? Oh, in May. Yeah, no, school is over in May. May 28th is the last day of school, but we still have, after May 12th, we have like final exams and whatnot. So the schedule is going to look different. But we'll follow this schedule for 2 weeks. Obviously, you all are all rotation A. For those of you who are in person, you'll be here Monday and Tuesday. Wednesday will be an entirely remote learning day. So everyone will be at home, and the schedule is a little bit different. So class will only be 30 minutes. This class will be from 8:40 a.m. until 9:10 a.m. And then Thursday and Friday, my rotation B people will be here. All right, so it's going to take some getting used to, but I think it'll be a good thing because it gets you all here a little bit more often, which I think will keep you engaged and give you the opportunity to take some better notes and ask better questions. So just wanted you all to be aware of that. Any questions about it? And for my people in person, please go ahead and take out your notebooks too, something you can write with. You don't have one? Okay, why not? You, you ran? Okay, all right. Sade, so you need one? Okay, I'll get one for you. What about you, Freda? You don't have one? Have you used one at all this semester? It's at home? Okay, so bring it tomorrow. What about you, Sade? Have you been using a notebook at all? Okay, so just make sure you bring it tomorrow, please. Okay, so of course we've talked about this a little bit, but March is National Women's History Month. I just want to remind you of that, and I was going to kind of bring up a story today, but we'll talk more about that tomorrow because I took too much time at the beginning of class, which I apologize for. Okay, let me get a notebook really quickly. Ooh, have your notebook. Go ahead. Cindy. Okay, so biotechnology. Biotechnology is an area of biology involving the use of living systems and organisms to develop or make products. So it places a specific emphasis on genetic engineering. So this is involving using things that are actually living, and you might manipulate them, as we'll talk about. You might change their DNA. You might inject them with specific chemicals. And the ultimate goal is to make a product. The product might be a medicine, it might be a type of food, it might be makeup, it might be an improved living organism. We'll talk about that. But this is obviously an emerging field of biology. And the moral questions that arise from this are important as well. Should we be doing this? Are human beings playing the role of God by tampering with DNA and tampering with how organisms, with their physical characteristics? These are important questions to at least ask. Okay. All right. So here's some products that have come from developments in biotechnology just in recent years. We've been able to establish new protocols for plant and animal breeding. Vaccines have come from the improvement in biotechnology, cloning mechanisms. And I would write down some of these examples if I were you, just to refer back to. You don't need to write them all down, but just to give you pretty concrete reminders about what biotechnology refers to. GMOs, those are genetically modified organisms. Typically, we refer to GMOs in terms of agriculture, plants that we could be eating. Pest resistant crops. Why is that important? What do we think about that? Why is it important that we develop pest resistant crops? What does that mean? Good, exactly. We don't want the bugs eating the plants that are meant to be eaten by us or maybe by domestic farm animals. So we can literally go in and tamper with the DNA of these crops such that they are either poisonous to insects or they just taste bad to the insects, and so they no longer want to eat them. So that was a big development that's taken place over the last few decades. Stem cell therapy. We've talked about stem cells before. Does anybody remember what stem cells are? What's special about stem cells? Okay, so stem cells are what we call undifferentiated. So a stem cell has no designated job yet. A stem cell is kind of like, if you're a fan of sports, what we might call a utility player, someone who can play multiple positions. They haven't yet developed into their role. You know, if we're talking about soccer, then this is someone who's, you know, fast enough to be a striker or a midfield player, but strong enough to be a defender. We're talking about basketball. This is someone who's tall enough to play in the post, but who's agile enough to be a guard. Stem cells are the same way. They haven't yet been given a specific role to play. They can develop into anything. They might eventually become muscle cells. They might eventually become nerve cells. So when we think about biotechnology, this is important because we can use these stem cells for people who maybe have had traumatic injuries that have paralyzed them. When a person is paralyzed, their nerve cells for a specific part of their body are no longer working. We can use these stem cells to produce to make new nerve cells. Naturally, our bodies do not make new nerve cells. Nerve cells don't get replenished. So if you suffer from some type of traumatic injury, those nerve cells that die are just gone. All right. So when you hear people say you're killing off your brain cells, that is kind of a real thing. Our body, with your skin, we can make new skin cells. With your liver, we can make new liver cells. With your muscles, muscle cells can be replenished. Nerve cells are different. Okay, so we rely on these stem cells to restore mobility to some people who may have been paralyzed. Gene therapy. Gene therapy is another type of biotechnology. So this is specifically for people who might know that if they have kids, their child might be more likely to develop a specific disease. So they will meet with what's called the genetic counselor and figure out how to limit the likelihood that they will have a child with the disease. Designer babies, we'll talk more about that. Lab grown meat. You guys have ever had the Impossible Whopper from Burger King, or there are other types of, what should I call it? It's lab grown meat essentially. Um, so that's an interesting topic as well. But biotechnology plays an important role in our lives, and it plays a role that you probably don't think about. The insulin, if you've got any diabetic people in your life, the insulin that they likely have to take came from bacteria. It was produced by bacteria, so that's a form of biotechnology as well. All right, so DNA fingerprinting, however, this is what we really, really want to talk about. So this is the definition that I'll ask you all to write down. DNA fingerprinting is the process of determining an individual's or a species' DNA. We talk about fingerprinting as, literally the fingerprints that are on the tip of your finger. But in this case, we're also talking more generally about just using DNA to identify an individual or to make a link between two species. So DNA fingerprinting allows us to establish links between biological evidence and a suspect in a criminal investigation. You all will explore this as a part of your assignment for today, but this is not a perfect process, and the technology is improving. It's getting more and more accurate and has gotten more accurate over the years. But there was a time when the DNA technology was very rudimentary. it made mistakes, and it eventually led to some people being incarcerated wrongly, being convicted wrongly for things that they did not do. And this is what you all will explore today. So this science is not perfect. And if you ever watch a documentary or you're ever, in court for whatever reason, you can actually listen to how these scientists will take the stand and they'll say, what's the likelihood of this happening? Or what's the likelihood that this is the same person who left that DNA? They won't say it's a 100% chance. They might say it's 1 in 10, there's a 1 in 10,000 chance that this is not the same person or that these two things don't match up. Cannot be 100% certain. But the technology has gotten a lot better. Okay, so we're going to look at a lot of things that look similar to this, what we're seeing on the screen now. What we've got is on the left, there's a DNA that was taken from the crime scene. There's a sample of DNA taken from the crime scene. On the right side of that dashed line, we see the DNA from 3 different potential suspects. We can use these DNA fragments to figure out who's the person who most likely left that DNA at the crime scene. Because the more fragments that the 2 samples have in common, the more related they are. We'll talk exactly about what these fragments are and where we get them. But clearly, in this case, the sample that was taken from the crime scene is most closely related to the sample from suspect 2. Cool. All right, so how do we create these DNA fingerprints? If I go back for a second, we see these different black fragments that are appearing in this gel. So where are we getting these fragments from? This is a process called gel electrophoresis. We'll talk more about what exactly this is, but it's a really, really cool process. You take a sample of DNA and you're going to place that DNA into what are called wells. You're going to load the DNA into these wells. And the wells are basically a mixture of what's called agarose, which is a sugar. That's why it has that OSE ending, agarose, and also mixed with the buffer. The buffer just kind of neutralizes some of the acidity of the DNA. You load it into the wells, and then you turn on this electrical charge. DNA is a negatively charged molecule. We know that opposites attract. So when I turn on this positive electrical charge, it's going to pull the DNA towards it. But the fragments, based on their size, can only move so far. So the larger fragments cannot move very far through this gel. They're going to stay at the top of the gel. They're going to stay close to the wells. The smaller fragments are more easily able to move through the gel. And so they move farther and farther and farther away. The end result is that you get this nice separation of the fragments based on their sizes. If the samples are the same, then the fragments should be the same size. And so you'll see the fragments lined up in the same ways if they're coming from the same sample. For example, I'll go back again. Oops. At the very top of these two wells from the crime scene and from suspect 2, we can see that there are kind of two fragments. Those would be the larger ones, the ones that aren't able to move through the gel very easily. The ones that get all the way to the bottom that have moved through the gel a pretty long distance, those must be the smaller fragments. And again, this is all coming from DNA. It's the sequence of nitrogen bases that's going to determine these fragments. Okay, so what can we use this process for? It's not just for crime scenes. Yes, forensics and forensic criminology, we definitely use this process a lot, but we can also use it for paternity tests, and we can also use it for cataloging endangered species. So we've got two examples here. And the example on the left side, we've got DNA from a crime scene. Which suspect left that DNA, most likely? This one. Suspect most likely left the DNA from the crime scene. Yeah, suspect number 2, because we can see that suspect number 2 has DNA fragments that are the same size as the crime scene DNA. That's why they're literally located. We can draw a line directly across. So suspect 2 more than likely left that DNA at the crime scene. What about in the other example on the right? Yes, suspect 2 again. Thank you, Sade. And the image on the right is typically what the actual DNA fingerprint would look like. Okay, so here's an example for a paternity test. Which individual here is most likely the father? Okay, let's see. Obviously, the mom and so some of the fragments. So let's just so the first fragment from the child could have come from dad three. Dad one doesn't have that. The second fragment, none of them has that one. Probably not mom yet that have this one. You move down further, only dad one has that one. And mom, dad one and dad three. So now dad one has two fragments in common, dad three has two fragments, and dad two only has one fragment. That one could have come from dad two or dad three. So now they all have two, but it looks like this last fragment probably tells us dad one is the father. It's not a great example because they're pretty similar, but it looks like dad one is most likely the father here. So again, we're just comparing, yeah, we're just comparing the location of these fragments. Oh, it looks like maybe we're saying dad three. Okay, I guess it could be. Okay. So here's the process of gel electrophoresis. You're loading the DNA into the wells. The longer fragments are located closer to the wells. The shorter fragments are located further away from the wells. They get pulled away. Remember, DNA is negatively charged. So when you expose it to a positive charge, the positive charge is going to pull the negatively charged DNA further away from the wells. Okay. So that we've got 15 minutes now. That should be enough time to knock out the exit ticket as well as the assignment. The assignment is about the Innocence Project, which is the video that I just skipped over. But go ahead and get back into Canvas now. Start off your quarter on the right note. Don't be the first person to miss an assignment. Set yourself up well and knock out this assignment really quickly. All right, it's called U6D1, the Innocence Project. There is also an exit ticket, so go ahead and knock both of those out. Use your time wisely. Brother Neftali, why don't you guys go ahead and knock out that work? Huh? I'm just saying, go ahead and knock out the assignment. Thank you. Good. What's that? You said you have one at home, right? So you can just go out the pages that you need. Did you finish? You finished the time? Why did you do it? You prefer to do it at home? Okay. So much is all fine. What about you, man? Did you do the assignment? U6D1, the Genesis project. Yeah. I just want you to actually use the time that we give. You know, I don't want you to feel like you have to do it at home and you're tired and you want to chill and relax. You might as well just do it during class. Get into the habit of doing that.
Please check your email and share the website of our credit recovery update to all of your students during all time credit recovery. Once again, check your email. There's a credit recovery update. Students only need to complete 80% of the course, 80% of the course with an actual grade of 60% to earn a credit. Review your email for details and have a great day.